you can make questions on the chat, but remember to do that just during the questions time so you don't bother the speaker. So now I'm gonna give the microphone to Pierre Lau. So welcome.
All right, cool. Well, thank you, EIS, and thank you everyone for being here and hosting this virtual symposium. Hope you're all staying safe and sane during this pandemic. But my talk is on honeybee nutrition. But I'm actually going to start it off with this kakapo parrot. This is a New Zealand kakapo parrot. It's actually endemic to that region, and it was extremely and critically endangered at one point. So these conservators are trying to rear them and get the populations to go up. So they're feeding them this supplementary feed. It's a very general feed for birds, but they weren't reproducing regardless. So they brought in two nutritionists for animals, and they applied this method called the geometric framework, which I would discuss on the next slide. And what they did was they compared their natural diet to the nutritional content of the natural diet to what the conservatory was giving them. And they found out that the protein and the lipid ratio of this natural diet was significantly different compared to the supplementary feed that was being fed. In addition, that supplementary feed was much lower in calcium than what was found in the natural diet, and calcium is needed for the birds to develop the eggshells whenever they're laying them. So as of pretty recently, these kakapo parrots, the population numbers have rebounded, partly because of the change in diets that they're able to make because of this type of research. So this geometric framework is known as a modeling approach, where we can actually model and explore how we can balance multiple nutrients at a given time in a nutrition environment. So in this example here, you have protein and carbohydrates on the y-axis, and this blue line is a diet that's very high in protein and low in carbohydrates, and this orange line is a diet that's very high in carbohydrates and low in protein. So in this example, this animal is consuming both of these diets so that it reaches this optimal intake target, so this red spot, where this animal will maximize its fitness or survival or reproduction, whatever its goals are at that given time. So I want to apply this model onto honeybees because bees face a lot of issues. This is a meta-analysis of a bunch of different papers throughout from 1989 to 2016. And in more recent times, I want to direct your attention to this orange bar here. I consider this to be nutrition, but bee nutrition has been significantly increased in terms of their interest for studying this topic. And one of the reasons for that is because the nutritional basis of pollen supplements is it's actually poorly understood, and a more rational approach based on honeybee ecology and physiology is needed. And that was from Dr. Jerry Wright, who is one of the premier researchers on bee nutrition. So this was a study that came out last year from University of Florida, and they compared different pollen substitutes that we currently have in the market to how bees performed over time. So you have time on the x-axis and the bee performance of a colony on the y-axis. So this purple line is a negative control. All of these other lines right here are different types of pollen substitutes that you can actually purchase and feed your bees. And you can see there really isn't a significant difference at all between the negative control and whenever beekeepers give these pollen substitutes available commercially. Sometimes they even perform worse compared to not giving them anything. Whereas natural pollen, wildflower, those bees actually perform best over time. So that tells me there's something wrong and mismatched with these diets. So my research objective has been to determine an optimal honeybee macronutrient ratio in these artificial diets. So I'm using this geometric framework approach for my methods. So for what I did is I had a field component, and we established colonies. These are nucleus colonies. These are small honeybee colonies consisting of 4 to 5 frames, and we caged them so that they cannot forage for anything else on the external environment, and we gave them a choice test and a no choice test. So I'm only going to talk about the choice test for the purpose of this talk because of time. But we gave them a choice between brassica and rose pollen. So brassica is mustard, and rose is like your wild rose or something in the almond family. And what we found is that bees had a very high preference for brassica compared to rose. And we had two treatment groups here that show us the same thing. In treatment A, we gave them twice as many arrays or dishes of rose compared to brassica. And treatment B, we gave them twice as many brassica compared to rose. So what this is showing is that regardless of what treatment it is, regardless of how many rose dishes there are relative to brassica, bees are significantly going after brassica pollen a lot more compared to rose. So they have a very high preference for this pollen type. And when you map this on the nutrient array here with the geometric framework, you have protein on your x-axis and lipids on your y-axis. We see that for both of our treatment groups, there is no significant differences in the amounts of protein and lipids collected. So that's just showing that there was no significant difference with both of these pollen types that were collected here. And the important number I want you to remember here is that the protein to lipid regulation was around 1.43 to 1 parts protein to lipids. So this ratio is actually really important. And when I first saw this, I was actually really concerned because a lot of the current knowledge is a 10 to 1 protein to lipid. So it's very high and biased towards protein. So when I got this such a low ratio, I was very concerned that I did something wrong or something happened throughout my experiment. So to confirm this, we brought this into the lab and we created artificial diets. And that's because pollen has a lot of other macronutrients, and there can be chemical cues and even odor cues and also visual cues that can affect a bee's preference. So we want to eliminate those preferences by creating our own diets in-house. So we created artificial diets with isolated soy powder, linseed oil as our lipid source, sugar as our carbohydrate source, and a bunch of other things I'm not going to get into detail with. But we created these different diets with different protein lipid ranges based on what we currently know about the levels of protein and lipids in bee bread and also pollen. And this is actually one of the first artificial diets that that's been created for honeybees, where we can actually manipulate the protein and lipid levels without diluting the other micronutrients in the diet itself. So we can do a lot of experiments with these PL levels. So we brought this in the lab and we got 1-day-old bees within the, we got 1-day-old bees and we collected them into cages in cohorts of 30. And we performed a no-choice test with 1 of 5 diets or negative control. And we also measured the bee physiology, which is total lipid content and hypopharyngeal gland size. Okay, so when we gave one of these 5 diets, so we have our diet types on the x-axis and the food collected on the y-axis, we found that the bees consume significantly more diet when they're given a 30-20 diet. And this graph here is showing the same exact results, just showed differently, but over time. Okay, so this is a 30-20, they're just consuming a lot more of the 30-20 diet. When you map this out on the geometric framework array, the protein on the x-axis and lipids on the y-axis, each one of these lines are nutrient rails, so the slope of the protein and lipids of the diets. The point I want to direct you to is these 3. If you look at the 3 intermediate diets, these bees would consume these diets up until it hits this point here, and it'll stop. This bee here will consume the diet up until it hits this point here, and it'll stop. So around 1.8 milligrams of lipids per bee, the bees would stop consuming that diet. So this tells us that there's some kind of regulation for lipids with these individual bees in the colonies. When we look at the physiology here, we saw that the bees given the 30-20 diet and these other 2 higher protein diets had significantly higher hypopharyngeal gland sizes compared to the other 2 diets. So this hypopharyngeal gland size is a gland that is that's indicative of nutritional status. So nurse bees use this to develop brood food to feed to the developing brood. And what this is showing is that these higher, this red line here is that this hypopharyngeal gland size is correlated to the amount of protein that was consumed. Unfortunately, we didn't see a significant difference with the lipids, and I think that's because the duration of the experiment didn't last as long as it needed to for it to actually see a difference. But we were interested in looking at lipids because the fat content is looking at the total energy stores of the honeybees. So now we performed a choice test here. We had 3 treatment groups with 3 different pairings of the diets within the cages. So in our first pairing, we had 35-15 paired with 30-20-30, and we found a significant difference between consumption. The second treatment group, we found no significant differences between the consumption when it was paired between 35-15 and 25-25. And in the third treatment group, we found a significant effect whenever the diet pairing was 30-20 and 20-30. The interesting thing is here, when we map this out in the nutrient array, we once again protein on the X and lipid on the Y axis. Both treatment 1 and 2 consume diets until they hit this 30-20 ratio line. And let me go back here. So for this treatment 1 and 2, neither of these diets were given this 30-20 diet to begin with, but they're both consuming them at the right proportions so they align at this 30-20 line. So 30-20, once again, that's if you convert it, when you do the math, it's actually 1.5 to 1 protein lipids. This one over here, 30-20 in a perfect world, in treatment 3, in a perfect world, the bees will only be consuming 30-20 and they wouldn't consume this 20-30 diet at all, but they were still eating this a little bit, so this diet was shifted over to the left. But the overall intake target that we see if you average them all out is 1.4 to 1 protein to lipid, which coincidentally is the same exact protein lipid ratio that we found in our field experiment, which I think is very rare. So to wrap some of this up, we have one of the first artificial diets that we created that we're able to artificially manipulate protein lipids. And from these diets, we're able to see lipid regulation in some of these intermediate diets. We didn't see any in the 35-15 and 15-35, maybe because the 35-15 was slightly too high in protein and the 15-35 was way too high in lipid content. So in the choice test, we found that bees regulate to 1.4 to 1, which is the same exact result as we found in the field with natural pollen. So what we found here was that the PL ratios from these field and lab studies show that there's a substantial difference in what we know about the protein lipid ratios currently known in literature. And really, it's not all about protein, as what a lot of these pollen substitutes suggest. There's actually a very important balance of macronutrients that we need to think about. So just some broader impacts here. This is just some of the examples of protein feeds in the market. And you can see that a lot of it's just very heavily focused on protein. You have like 56% protein, 3% lipid. So lipids are not really paid any attention to at all in the industry. And right now, we're really expanding our work right now with looking at how pathogens affect these diets as well, or how bees affected with pathogens can influence their preferences or the tolerance to these pathogens. So we're giving bees these different diets and seeing how they can help bees tolerate pathogens. So we're looking at how nutrition can help these bees with the form wing virus here and Nosema ceranae, which is a microsporidium parasite. So just a really quick plug. We do have funding for the summer, so if there are any undergraduate students here that might be interested in getting involved, we might be able to get you on, depending on this whole pandemic and whether or not it will die down a little bit. But just reach out to me, send me an email, or just send me a message on chat if you're interested. But to quickly acknowledge everyone that was involved and the funding sources, these are my advisors and collaborators. We had other undergraduate assistants as well. And our funding sources were from Louisiana Beekeepers Association and the Eastern Agricultural Society.
Okay, thank you, Pierre. Now we're gonna have 3 minutes for questions. As I told you before, you can unmute yourself to make questions, or you can use the chat to make questions.
No questions?
Yeah. Oh, someone made a question.
Oh.
What made you want to study bees?
So that's a good question. I just kind of really stumbled upon it. When I was an undergraduate, I started off at a lab studying algae biofield ecology. And that summer, when I went home, I reached out to a local college or university, and there happened to be a bee lab that studied wild bee pollination on watermelon agroecosystems. So once I got sucked into that, once I started on that, I found out when I went back to school, there was a honeybee lab. I got involved there. Just got really sucked in. The more I learned about bees, the more fascinating I thought they were. So I just got really stuck into this whole um, loop of being very interested in them and just learning. With the more I learned, the more interested I became.
Okay, does anyone have any other questions? Mm, yeah, because it's almost time for the, okay. Tyler asked, do you think there are broader applications for your diet to the beekeeping community?
Yeah, so I think there are definitely broader applications for diet. Before earlier this semester, we actually started a preliminary study with um, two undergraduate students where they were testing this diet out in the field and how effective it can actually be. So the reason why I didn't get into too much detail of the diet is because we are thinking about going for some intellectual patent or property rights for it so it can be further developed. And so that the beekeeping community can get this knowledge and use it and modify their diet to help improve the bee nutrition.
Okay, thank you, Pierre. Now we're gonna wait for the next presentation that will be at 10:20. Thanks. So now we're gonna have Chris Wright presenting about sugarcane aphids. So I ask you to use the chat just during the questions time, and you can unmute yourself too if you need it during questions. So thank you for coming, Chris, and I hope you have a good presentation as always.
All right, thank you. All right, so let's get started. Today I am going to talk about the sugarcane aphid and essentially how diet affects what happens on the other side. So let's get started. This is the sugarcane aphid, Melanaphis saccharide, and this aphid is a parthenogenetic species, so it reproduces asexually. As you can see from this figure, the adults can either be winged or wingless, or alate or apterous. The sugarcane aphid is a pretty important pest of grain sorghum. This map is a distribution of the aphid throughout southern United States, and you can see it encompasses a lot of southern and southeastern grain sorghum growing regions. It can also go as far west as California in some cases. So this is becoming a growing problem for sorghum producers. If anybody has not seen sorghum before, this is what it looks like. It is primarily used as an animal feed crop, and under healthy conditions, you're going to see a lot of kernels that are building, and the plant is going to look, this is white sorghum, so white and vibrant. The problem is once you get a lot of aphids on the plant, depending on what growth stage they attack, you can see something that looks like this, where I want to call your attention to the background where these kernels, which were supposed to be full, actually look almost blackened and moldy and are very sparse in between. Whereas in the foreground of this photo, the aphids are populating this leaf in the hundreds and probably in the thousands on the plant. So any sorghum producer is going to tell you that this is an issue. And one of the main things I want to highlight for this talk is the fact that aphids produce copious amounts of what's called honeydew. Honeydew is this waste, the sugar-rich waste product that occurs after the aphids pierce into the sorghum leaf and suck up what's called the phloem, that's sugar-rich tissue. Honeydew, as you can see, is quite sticky and it coats the entire surface of plant leaves. And this can lead to molding and additionally light. And if plants can't get light, they cannot photosynthesize. So directly their growth and development is hindered. Again, growers would say that honeydew is just all around a bad thing and it's a terrible thing to have on your plant. But I'm always interested in figuring out both sides of the story, and I hope to show you throughout this presentation that honeydew is almost a double-edged sword. And I say that because some amounts of honeydew can act as an attractant to natural enemies. And by natural enemies, I mean either parasitoids like this wasp stinging an aphid here, or predators like this syrphid or hoverfly. In some cases, either parasitoids or predators can spot honeydew and use that as a breeding ground to lay their eggs, or directly located or associated with host populations to start attacking or ovipositing inside of those hosts. Now, this is pretty great, but since we are talking about biological organisms, I do want to note that honeydew sometimes can attract a predator or parasitoid differently, depending on whether it is on one plant versus another, even if it's the same pest species occupying the plant. So for example, let's talk about this hoverfly. This is the marmalade hoverfly, and it is known to attack a pea aphid. And pea aphids, for anyone who knows, they can have a fairly large distribution of host plants. For example, they are known to attack these two plants. The first one here is red clover, which on this graph is going to be this dark gray color, or broad bean, which on this graph is the light gray color. And depending on what plant species they are on, they can produce different amounts, total amounts of honeydew. As you can see on red clover, pea aphids produce a lot more than on broadbean. And as it turns out, this is directly correlated with the oviposition, the egg-laying rate of this hoverfly predator. So as we can see, there is a clear preference for laying eggs, which will start to develop and start eating the associated aphids on plants where more honeydew is present. So for me, I think that's very interesting and relevant to my research because grain sorghum is not the only host plant for sugarcane aphids. They can also occupy this weed called Johnson grass. Johnson grass is going to be located in all areas where sorghum is grown, and oftentimes it is in close proximity to the crop, which means there's ample opportunity for sugarcane aphids to occupy both host plants at around the same time. And that's important to me because I have been researching in the Texas A&M farm for a couple of years now, and I've noticed that there are about the same number of aphids in both grain sorghum and Johnson grass. However, and for some reason, the number of natural enemies, that again is the parasitoids and the predators, is a lot more abundant in grain sorghum than it is in Johnson grass. And for years, I didn't know why that was. It didn't make sense to me. But after looking at honeydew and given what I've just shown you, I think one of the reasons a difference in the amount of honeydew that is produced on either of these plant leaves, and could also just be the type of honeydew that is present or the quality of honeydew. So that's really what my research pertains to today. I wanted to see, is there a difference in sugar concentration and also the identities of individual sugars in honeydew between grain sorghum and Johnson grass-fed sugarcane aphids. I also wanted to take this project a step further by looking at sugar concentration and composition over time. And the reason I'm interested in the time aspect is because I know that in some aphids, like the cornleaf aphid here, there are cases where sugars, the percentage of sugars in a honeydew sample can change over a period of weeks. For example, in this graph, the x-axis represents individual sugars in a honeydew sample, and the y-axis is the percentage of those sugars compared to of the sugars. And I want you to focus primarily on glucose and sucrose here to show that with glucose, while the concentration or the percentage of sugars remains relatively the same for the first 2 weeks, after 3 weeks, there seems to be a spike, and almost 45% of the total sugars is glucose. Whereas with sucrose, I see, again, more or less the same amount until 3 weeks, where that amount of sugar really just decreases significantly. And there's a possibility that that could have happened with sugarcane aphids on either host plant, and it's something I chose to explore. So here's what I did. This is my experimental design. I used a clip cage with 10 clonal aphids. Again, the sugarcane aphid produces asexually. And then depending on the species, I placed that clip cage to a sorghum or Johnson grass leaf for a period of either 24 hours or 72 hours. And then once I collected the honeydew, I used the technique called high pressure liquid chromatography, or HPLC, to essentially determine how much of certain sugars and total sugars were in a particular sample of honeydew. And these are my results. This graph is showing you the total sugar composition, abundance in grain sorghum versus Johnson grass. And on the y-axis here, this is the peak area, which is an HPLC unit that serves as a proxy to concentration. And I'm going to talk about why I use this instead of actual sugar concentration in 2 graphs from now. But the point of this graph is to show you that these are box plots because I want to indicate that there is quite a variation between my samples. But looking at the average, which is this white bar here, on average, Johnson grass has more total honeydew sugars than grain sorghum. And this occurs irrespective of the time, whether it is 24 hours or 72 hours, you still Johnson grass is going to have a slightly but significantly greater abundance of sugars than grain sorghum. So yeah, that's what's occurring. Now let's see what happens when you are breaking everything down into individual sugars. And that's what I've done here. I have compiled 6 sugars that I've seen significantly in each of the samples. And those sugars are fructose, glucose, maltose, sucrose, and these unknown sugars. And that, again, is the reason I have this peak area instead of sugar concentration, because I don't know what these are. And if I don't know what these are, I cannot use known sugar standards to create a calibration curve to essentially figure out that concentration. Fortunately, peak area is a good substitute for this. The point of this graph, however, is to show you that when you look at individual sugars, the fact is that Johnson grass, the variation of sugars in Johnson grass is not significantly different from the variation of sugars in grain sorghum. And by variation, I mean all the differences in each individual sugars here. However, there's this one sugar, this unknown sugar, I really need to figure out, which for some reason is a lot more abundant in Johnson grass after 24 hours than it is in sorghum. And I'm so happy I decided to do this after 24 and 72 hours, because after 72 hours, that difference is gone. There's a complete homogenization of these sugars, of all of the sugars in the sample. And that really makes me curious as to why that's happening. What is going on with this particular unknown sugar? So in summary, what I found, first, I know that the total sugar concentration is going to be slightly greater in Johnson grass. And the composition and concentration of most sugars is not going to be significantly different between plants and over time, save unknown one sugar, which for whatever reason is more abundant in Johnson grass after 24 hours, but then reduces in proportion to other sugars in uh, grain sorghum. So it makes me think, now that I know natural enemies are more apparent on grain sorghum than Johnson grass, it makes me want unknown sugar acts as a type of repellent, at least initially, and that could lead to a difference. So that brings me next to my next steps. First, I need to figure out what that sugar is, what's going on there. And second, I want to conduct some natural enemy choice tests, especially in Johnson grass honeydew for 24 hours and 72 hours, which will hopefully let me see if there's any effect of this unknown sugar on natural enemy preference. And with that, I'd like to thank everyone for helping me with this presentation, and I'm happy to take your questions.
Thank you, Chris. Now we have a time for questions. So you can unmute yourself or make the questions on the chat. So Chris, I was thinking, you're gonna use like the honeydew as somewhat to attract natural enemies or not?
I am, yeah. So my idea for the no choice test is to have a disc of honeydew from aphids fed on Johnson grass and aphids fed on grain sorghum, put them in a petri dish, and just see where the natural to figure out if they are preferring one honeydew over the other. And it's so important for me to figure it out, especially with Johnson grass after 24 hours of collection and 72 hours, to simply see if there is a striking difference in preference between those time points.
Okay, but are you gonna make like some artificial attractants or not?
So what I still have those honeydew samples that I collected, and I plan to have, they're all stored in the fridge, so I plan to use those as a choice test. But I also, yes, I do plan to have some artificial diets made, particularly those with that unknown sugar, once I figure out what the identity is, just to see how natural enemies can fare on that particular sugar.
Okay, but this, like, I don't know, the solution that you're gonna make, it's not gonna have like the same sticky feature that has the real honeydew, right?
Mm-hmm. Yeah, so with that one, that's a good point. And for me, what I may be, of course, we're still working the methods for that, but we may have to do something that can stimulate that sticky condition. Or what we could do is feed them that solution, but also feed them those actual honeydew diets just to see overall what's happening. Do they survive better on honeydew versus an individual sugar? Survive better on water, which is a control, not likely, but yeah, all of those different factors I have compiled in a potential experiment.
Okay, thank you. Does anyone have any other question?
Yeah, I have one small question. Hey, maybe like I missed out this point. So whatever the analysis you did, it's on the honeydew, right?
Yes, so on the analysis using high pressure liquid chromatography, I
Yeah.
basically looked at the amount, the concentration of total sugars and also the individual sugars, those fructose glucose amounts for the samples in Johnson grass or grain sorghum.
What about the sugar content in the plant itself before the honeydew? Maybe like the unknown sugar, what you're saying, is it just like transported from the plant, which is different between the two plants?
Yeah, that's a great question. I wanted to separated from the plants to the insects to the natural enemies. And then I spoke to my committee and they said, you won't graduate in time if you do that. But
Is it, I'm just curious whether this is present already in the plant, just like it's not digested in the insect and it got like excreted as a honeydew, or like how it is. Like, I'm just curious about that. Is there any
those
way
sugars.
yeah, trace back to see the differences are there already in the plant itself innately?
So I think that's a really good question. And I do think there is one person that is actually looking at the individual sugar composition in grain sorghum. Uh, what I do know is that honeydew, based on my review of the literature, actually tends to have more. So in plants, they tend to have glucose, fructose, and sugar. And honeydew tends to have more oligosaccharides like malezitose, for example, which does suggest that they are combining those inside of their gut or inside of whatever in their
Yeah.
body to have those sugars that are not going to be present in that state on their plants. Does that answer your question?
Yeah, exactly. Something on the same line, but I'm also just curious, is there any precursor or anything where we can trace back? Whether like
Yeah.
the basic question I have is like whether this difference is inherently present in the host plant itself, so that like you see the differences in the honeydew. That's
Hmm.
yeah,
So
but
yeah, I would estimate that, at least based on what I've read, at least with malezitose, which is an oligosaccharide, that you're not going to find that in the plant, at least in the concentrations that I've found that in the honeydew.
okay, cool. Thank you.
Mhm.
Okay, thank you, Chris.
Yeah. Fine.
I'm gonna wait 1 minute more, but now it's my time to talk. And let me see, someone's talking. No. Okay, let's gonna start. My name is this huge name that you can see on the bottom, but it's easier if I say that my name is just Janaina Cunha, and I'm gonna present today the presentation titled "Can Cotton Associate Fungi Affect the Behavior of an Aphid Predator?" So to start with, I'm gonna make a background a little bit about some stuff that I think it's important for you to know. So the first thing is the cotton crop that I used for my studies. And the species that I used was Gossypium hirsutum that belongs to the Malvaceae family. And it's known as the upland cotton. And it's the species most widely cultivated around the world due to its high yield. It is one of the most valuable fiber crops in the world. That's why it represents a large part of the global socioeconomic sector. As any other crops, cotton is attacked by a range of pests that can have negative effects on production if not controlled. So that's why the economic and ecological sustainability of cotton production is directly related to the efficiency of combating agricultural pests and minimizing adverse environment effects. So the first organism that I'm going to talk about is the cotton aphids. And the cotton aphids or the melon aphids, it's one of the most destructive pests in cotton. and it's widespread around the world with a broad range of host plants and can cause serious economic losses in cotton, mainly because of its rapid life cycle and asexual reproduction that enable them to reach huge populations in that short period of time. The aphid infestations in cotton can directly or indirectly damage the plant. The direct damage is because they consume the phloem sap, and this will decrease the plant growth and development. And the indirect damage is due to the aphids being vector for virus transmission and contamination of mold uh, promoted by the honeydew. The broad spectrum insecticides are widely used to control aphids in cotton, but we can see that the insecticide resistance is increasing, and the impact of these aphids of the crops highlight the importance of studying the natural enemies that can be used to control this pest, as we saw in Chris' talk before mine. So the natural enemy that I use it for my studies, it's the predator, the convergent lady beetle. The convergent lady beetle, it's one of type of the key natural enemies that we can find for cotton aphids. Chris mentioned to you to parasites. We can find spiders too. And this species, the Hippodamia convergens, it's the most common Aphidophagous species native to North America. We can see that the females, they may lay from 200 to more than 1,000 eggs in a period of 1 to 3 months. And after hatching, the larvae may eat between 30 to 50 aphids per day. Thus, because of their predaceous habit and distribution among crops attacked by aphids, this species is a key part of many agroecosystems and an important biological control agent. The last thing that I will talk about on my background is the plant-associated fungi. We can see that the phytobiome is the association between plants, environment, and micro and macroscopic organisms. These individuals, the microbes, they can influence the plant growth, the health, the productivity, and may also be manipulated as a key tool for insect pest management, IPM, in agricultural systems. We saw already for some publications that these microbes can be used to increase the natural enemies efficiency and to control the insect pests. They can have this dual life. Several studies have shown that fungal endophytes enhance plant resistance or tolerance against biotic and or abiotic stressors. A biotic stressor can be the pest, as we're talking here. So some species haven't shown potential for insect control because they can affect the pest reproduction, fecundity, survivorship, development, and host selection. So the objective of my research, it was to investigate the effects of plant-associated fungi applied to cotton plants on the behavior of the predator, that it is the convergent lady beetle. So we know that some plant-associated fungi has effects on the aphids, like decreasing the reproduction 50%, but we don't know if these microbes will interfere with the natural enemies, like I don't know, maybe the aphids will have a different quality as a resource for the lady beetle. The first thing for my methodology was doing the seed treatments. So first I did a spore suspension, and I used 2 species of fungi that we have in our library in the lab. It's Physalimon flatum, that we know as, I will call 490, and Ecatomium globosum, that is the 520. Before apply my spore suspension, I need to sterilize the seed's surface. And after that, I will inoculate the seeds with the suspensions and a sticker solution that is methyl cellulose. The seeds will dry and I will plant them in the greenhouse. For the insects, for the aphids, I infested plants with cotton aphids 2 weeks prior my behavior trials. I commercially acquired the lady beetles, both females and males. After that, I need to sex them to make sure which sex I have. I starved them for 24 hours prior the behavioral trials. And these beetles were placed in cups inside an incubator with controlled conditions. For my white tube olfactometer, I had 2 mason jars that it was my chambers that I put the plants inside. I had the air coming for a pump that went inside these jars and outside for the tube, so it can go inside my white tube. I introduced a single lady beetle of both sexes for sure at the base of the white tube, and I let them acclimate there for 5 minutes before release them on the white tube. I assess the behavioral responses from these stimuli emitted by the air coming to the plants. And I consider a choice when the lady beetle entered at least 3 centimeters of the Y arm and stayed there for 20 seconds. So the numbers that I used for my trials, it was a total of 40 individuals. So my first trial, it was, sorry, the control with aphids versus the treatment, the fungal treatment, that it was or 490 or 520, so both plants with aphids. My second trial was comparing control and the fungal treatment, but both plants with no aphids. In my third trial, it was using the same treatment, like control versus control, but one plant with aphids and another plant with no aphids. I used the same amount of females and males, and I recorded the data, my variables that were first choice, latency, and residence time. So for my results, as you can see it in this table, it's the first choice. When I was comparing the control, aphids versus no aphids, the males were having a significant choice for the plants with aphids from the stimuli emitted by the plants infested with aphids. When I was comparing the 490, both one plant with aphids and the other plant no aphids, I was having a different thing for the female. The female were choosing, having her first choice, the plants with no aphids. For my latency, the thing that I think it's important to mention, here in this graph, you can see it on the x-axis, is the time for the lady beetle to make a choice in seconds. In my y-axis, it's the density. And you see it that when I have the fungal treatment 520, the females were making a faster choice here. They're taking a less time to choose the plants with the fungal treatment, regardless the presence or absence of aphids. For residence time, when I was looking for the control plants, that it's the plants that doesn't have any fungal treatment, I see it that both males and females were staying more time in association with the stimuli emitted by plants infested with aphids. So my conclusions are that although we can see that the fungal effects were minor, we see that there is some effect that can affect the multi-trophic interaction between plants, herbivore, and their predators, and for sure the microbes that are insert in these interactions. The attraction of more natural enemies to the plants treated with the fungi could provide another means of reducing herbivore populations and might maintain the pest population under the economic injury level. So the significance that I think for my work is that microorganisms can be used in IPM to inhibit pest activity and are making them more vulnerable to predation. And we know that we need more environmentally friendly practices so we can minimize or eliminate the past impacts on cotton, and for sure helping the economy and promoting cotton sustainability. So that's why I think it's critically important to understand if the manipulation of microbes associated with crops not only will affect the crop pests, but the natural enemies as well. So for that, I want to thank CAPS, that it's a Brazilian government company that funded me and my lab mates. Now I'm open for questions. Oh, sorry. You can both unmute yourself or make questions on the chat. Okay, if anyone doesn't have any questions, I will stop the meeting. So if you have any questions, it's the time now. If you don't, it's fine too. I will just end the meeting.